This afternoon, we've chatted about innovation in drug treatment. We've talked about
Cet
the
après-midi,
future
nous avons
of
dit
academic developments. And now we're going to talk about beyond the drug innovation. And in this session, we welcome two trailblazers to discuss their innovative work, the thinking behind their work, and the implications for future cancer treatments based on the work that they're doing. We are very privileged to welcome Dr. Andrea Cercek, who is a medical oncologist specializing in the treatment of patients with gastrointestinal cancers, most particularly colorectal cancer. Her research is focused on the development of novel, systemic, and regional therapies, including approaches based on tumor genomics, with the goal of improving outcomes while minimizing treatment-related toxicities. She's also the founder and co-director of the Center for Young Onset Colorectal and Gastrointestinal Cancer, the first clinic of its kind in the world. We also welcome Dr. Rebecca Auer, who is the scientific director for the Cancer Therapeutics program at the Ottawa Hospital Research Institute. She's an international leader in surgical oncology and translational research in surgery. She's also professor in the Department of Surgery and the Department of Biochemistry, Microbiology, Immunology at the University of Ottawa. Dr. Auer holds a Tier 1 clinical research chair in perioperative cancer therapeutics and leads a translational research program focused on understanding the promotion of metastatic disease in the perioperative period following surgical stress. Dr. Auer has led four translational clinical trials of perioperative cancer therapies based on this preclinical research. Welcome to both, and thank you for being with us today. Andrea, I'm going to go to you first, and I would like you to spend just 5 or 10 minutes talking about the work you're doing, particularly in some of the very innovative spaces. You're well known for a paper which has been recently published, some of your thinking behind that work, and how you feel it's defining the future of research in cancer care going forward.
Absolutely. Thank you so much for having me. So as you mentioned, I specialize in colorectal cancer, and I have a particular interest in early onset colorectal cancer. So that's in individuals under the age of 50, where the incidence of colon and rectal cancer, but particularly rectal cancer, has been rising steadily for unknown reasons, really, and it's a global phenomenon. And the goal of our center is sort of twofold. One is to help patients navigate this disease from diagnosis, treatment, and into survivorship. But the second goal is really to focus on research. And one piece is to identify the etiology behind this rise. And then the second is to improve treatments for patients that are diagnosed with colorectal cancer. And specifically in rectal cancer, I've had an interest in quite some time in trying to optimize therapy so that we can increase responses, improve outcomes, so meaning improve cure rates, but not only cure rates, but potentially improve outcomes so much that we can minimize the toxicity of some of our curative treatments. And what I mean by that is that rectal cancer, because it's located in the pelvis, is treated really with 3 different therapies. It's treated with chemotherapy and radiation, and then surgery. And we do well with that trimodality therapy in terms of curing patients, getting rid of the cancer completely. But our treatments, and in particular radiation and surgery, have a lot of toxicity. So about 30% of our patients with rectal cancer need a permanent colostomy or permanent bag. Radiation has a lot of negative effects on bowel function, bladder function, and particularly in our young patients, sexual function and fertility. Because the ovaries and the uterus are in the field of radiation, often women are infertile. It affects also fertility in men. And so while patients are cured, especially as we're seeing more and more younger patients, these issues are becoming increasingly more important. And so my research really is focused on trying to optimize this treatment, or do what's called neoadjuvant or preoperative therapy, to try to potentially omit surgery. And this is not unique to us at Memorial. This has been done worldwide and has really become an increasing interest in rectal cancer research in particular. And what we did specifically at Memorial is the idea of, can we find certain groups that will respond better or that will respond really well to a certain therapy, treat them, and then see how much we can really benefit from that therapy and potentially omit radiation and surgery. And when we're treating these patients, we notice that patients with mismatch repair deficient tumors, or what are called MSI high tumors, that just have the tumors and have a different genetic makeup. So there's a lot of mistakes that occur when a tumor is mismatch repair deficient. So it looks different to the immune system, and the immune system is already engaged in fighting it. But what we know about these tumors is they're not that sensitive to chemotherapy. And we noticed that when we were treating our patients with early stage rectal cancer, where, as I mentioned, we would really try to optimize therapy to omit surgery. So we gave them chemotherapy up front, and we noticed that most of the patients did really well with chemotherapy. The tumors responded very nicely. But this group that were mismatch repair deficient or MSI high, up to 30% of them actually had growth on chemotherapy, which we were not used to seeing at all. And so when they had progression or tumor growth, or when they had recurrence of tumor, we borrowed from our knowledge in advanced disease that these tumors were very sensitive to immunotherapy. So when we gave them immunotherapy, they responded really well. So the idea brought about by these patients that we really had to treat because they didn't do well with our standard therapy was, what if we designed a trial where instead of chemotherapy, we used immunotherapy up front? So these were early stage rectal cancers who normally would have had chemotherapy, chemoradiation, and surgery. And our trial said, okay, let's try immunotherapy with a PD-1 blockade, a drug called dostarlimab, an anti-PD-1 drug. And let's give that drug for 6 months. Watch the patients really closely on the immunotherapy, and then pause and evaluate with imaging, a rectal MRI, evaluate with an endoscopy, like a colonoscopy, but just a peek at the rectal tumor, look at the response. If at that point there was no tumor left, then the patients could be enrolled on what we call the non-operative management protocol, meaning that they didn't need any further treatment. They would just be followed really closely. If they had disease still after the 6 months of immunotherapy, then they can get the standard of care chemoradiation. And then again, they had the opportunity to just be followed if the tumor completely disappeared after both immunotherapy and chemoradiation. And if the tumor was still there, then they would undergo surgery. So this way, they could have had all the standard treatments, but we wanted to give the immunotherapy an opportunity to work to see just how much of those treatments we could potentially omit if we really hit a home run with immunotherapy. And what we saw, which was just, absolutely incredible, and as I've said before, an oncologist's dream, is that every single patient that we treated that received 6 months of dostarlimab, 6 months of the immunotherapy, had no tumor left after the 6 months of treatment. So no one has needed radiation so far, and no one has needed surgery. So this has really been, incredible, most importantly for our patients, because they didn't need the radiation, they didn't need the surgery. They did great with the treatment. There was very little toxicity. They were left feeling just completely normal. Most of them said, I haven't felt this good in years, probably because they had some symptoms of the rectal cancer. So absolutely no toxicity from the treatment, back to living their lives feeling fantastic. And then it was really exciting for oncology and for science, because it was the first time that we're able to replace a definitive treatment, which usually involves at the very least chemoradiation and surgery, with just a systemic or just a therapy like an immunotherapy to completely eliminate the cancer. And so that piece was just, incredibly exciting for us. We are continuing the study. And then, of course, the key questions are, why are these tumors so sensitive? What is different about this early stage disease, the environment in which the cancer sits, that makes them so exquisitively sensitive to immunotherapy. Because as I mentioned earlier, this treatment works when the cancer is more advanced, like stage 4, but it doesn't work as well. So there's something different. So there's obviously a huge research interest now to try to identify what that is and then learn from it, and of course expand it to treat patients beyond those that are mismatch repair deficient or MSI high. And then additionally, what we're doing, which is really exciting, is that MSI high or mismatch repair deficient tumors are not unique to the rectum. They involve the colon, really any solid tumor. So tumors of the esophagus, the stomach, the pancreas, liver can be MSI high. So we've expanded the study now to any patients whose tumors are mismatch repair deficient or MSI high and are early stage, with the goal, of course, to try to replicate this and try to give them just immunotherapy to be able to eradicate the tumor, as opposed to a combination of standard treatment like chemoradiation and surgery. So really excited about the future, obviously thrilled, and looking forward to more data and learning more from this therapy.
It's an astounding home run, and certainly took a lot of place in the medical press, and the study certainly has had a lot of profile. I do have one question for you, which is, when you talk about it, it's how did you even know what the right questions to ask going in? How did you choose the course you did? Because it took you to such an innovative place. Your passion for patients is very clear, but why that course?
Yeah, it's a great question. I think it came really from the data, what we knew in advanced disease, because we knew that there were some of these MSI tumors that are very, very sensitive, colorectal tumors that were really sensitive to just immunotherapy alone. So the patients had chemo, they had, all kinds of chemo, and then they got the immunotherapy, and many of them responded and were cured. And then it came from our experience in rectal cancer, and really the patients whose tumors we watched grow on our standard treatment, or even after chemo, radiation, and surgery, the tumors grew back. And then we saw that when we gave immunotherapy, they had these beautiful responses. So the idea was, it was twofold. It was sort of, can we optimize this, but how much can we get away with, And I think that was the part where maybe, just being an optimist, I thought, why not? And I certainly, have to say, I relied very much on my colleagues in radiology, in radiation oncology, and particularly our colorectal surgeons, who said, okay, yes, we're on board with this, and we can call a complete response. We can say the tumor's completely gone, and yes, we feel comfortable watching the patient very closely every 4 months on this non-operative management protocol to make sure that it doesn't come back. So it was certainly not, a solo effort. This was 100% a tumor board effort, and really, absolutely necessary. But I think it was a little bit of, optimism and just kind of what we knew from science that led us to kind of this design.
Mm-hmm. I love how you said that optimism is the path. Thank you so much, and congratulations on a wonderful result. Rebecca, I'd like to turn it over to you now to talk about some of the research work you've been doing, some of the innovation. I know you've got some slides, so Chad, if you could help us with that would be tremendous. Thank you.
Thank you so much. That was definitely an inspiring first section there. I'm going to talk about a clinical trial platform that's sort of at the very opposite end of that clinical trial spectrum. So completely different from the offering, a very novel therapy in a very new way, sort of experimental. This is definitely a little less sexy, but I think comes from the same spirit of wanting to make care better for patients and making sure that they have the best options for care possible. So this clinical trial platform I'm going to talk about is called React NSQIP Remote. We sometimes call it the R&R platform. And really pleased to be talking about it here on behalf of a whole team of researchers and oncologists. So as everyone knows, there's a lot of challenges to conducting randomized controlled trials in general. And of course, I'm a surgeon, so I'm particularly interested in randomized controlled trials in patients who are having surgery. and that time around surgery, which we call the perioperative period. We know that overall, less than 5% of patients participate in clinical trials in Canada. I think the stats are actually somewhat similar in the US, and even fewer surgical patients participate in trials. And so that means that patients aren't getting the opportunity to participate in trials and understand whether they're beneficial. We know clinical trials are very expensive. They require a lot of infrastructure, like an REB, clinical coordinators. So at some of the smaller non-academic sites, it's very hard for the clinicians there to have their patients participate. And outcomes is quite variable in Canada and certainly in the US as well. And surgical morbidity, so complications after surgery, can be very expensive. So next slide, please. One of the innovations that was not created by me, but created by a statistician, Dr. Dean Ferguson, and an oncologist, Dr. Mark Clemens, was the Rethinking Clinical Trials platform, and we call it the REACT platform. And what it's really designed to do is, as I said, evaluate standard of care therapies, like which drug is better for a certain condition, which treatment do patients prefer, that kind of thing. So there has to be some clinical equipoise there. And next slide, please. As discussed, traditional clinical trials, they can be quite cumbersome. You know, the doctor engages the patient agrees, and then a research coordinator then comes to see the patient. They sign often a 20-page consent. Then the treatment is administered, usually within a hospital, sometimes with extra visits, and then the coordinator comes back and collects the various data points. And this is really important for early phase trials, to have all those checks and balances and all that safety. But for later stage trials, the REACT platform was designed to break down some of these barriers. So it's based on the concept of integrated oral consent. So patients don't sign a consent. It's simply documented in the chart that a consent discussion happened by the clinician. They're randomized right there, real time. The clinician can do it on their phone or their desktop computer. They're not for blinded studies, so the patient knows what treatment they're randomized to. And then the data collection is done through a common data infrastructure. And in our case, we're using a surgical perioperative or postoperative morbidity database, which I'll talk about. So really, it's supposed to be a streamlined process. It's more cost effective. It really embeds that clinical trial in the clinical care. Patients sometimes don't even completely realize they're in a trial. They know that they have been randomized to one of two treatments, but normally which of those treatments they got would probably simply depend on who their surgeon was or who their oncologist was. And so these have real world generalizability. So the REACT platform is very successful. Next slide, please. Actually, if you can pass the next one. The next slide. So this platform's been very successful. Over the last 8 years, there's been 23 REACT trials and 4,100 patients accrued. So really a large number of patients in a very short period of time when you consider that this is a run out of a single site. Next slide. So our question was, can we use this platform and ask questions about postoperative morbidity, so complications after surgery? Next slide. So why would, I'm a cancer doctor, why would I care so much about postoperative morbidity? But we know that things like surgical site infections, and here's an example here, a wound that the abdomen that is quite red and swollen and sometimes has to be opened up, as you can see in the other picture. And they're associated with worst outcomes. They're quite common, and they are costly to the system. Next slide. And here's just some stats from the WHO, but you can see this is very serious, and they cost a lot of money. A lot of people get them, and it's the most frequent complication in surgical patients. So what does this have to do with cancer? Next slide. In pretty much every cancer that we know of, including colorectal, surgical site infection is associated with the worst cancer outcome. So this is just one paper, for example, showing a 30% decrease in relapse-free survival when patients got a wound infection or a surgical site infection. Next slide. And there's a number of studies. Here's something called a meta-analysis where they look at all studies combined, basically showed that patients are 1.4 times more likely to die of their colon cancer if they have a surgical site infection. So this actually does matter to patients and to their cancer outcomes. Next slide. And actually, we study this in the lab. We study how surgery impacts on cancer recurrence. And from the graph there, you can see that this is an animal model. And when the mice have what's called a laparotomy or an abdominal surgery, they get increased metastases to their lungs of colon cancer. And then when we have an infection in the wound as well, then they get even further increase. And actually, we've been able to show that this has to do with the immune suppression, so the fact that the immune system is not working well after surgery. So next slide. So we wanted to see whether we could merge this REACT platform with a data collection, a perioperative, postoperative data collection service that the American College of Surgeons has called NSQIP. It's called the National Surgical Quality Improvement Program. And many hospitals across Canada and the US, hundreds and hundreds of them participate in this program. And basically the hospital, through a very standardized way, collects risk-adjusted 30-day surgical outcomes for patients. And it's done by trained surgical clinical reviewers. And it allows us to benchmark our hospital and our care across all of North America. So this data is available. Could we use this data and run a practical or pragmatic clinical trial? Next slide. So, for example, one of the questions that many surgeons don't know the answer to and have a variety of different practices is what preparations should patients with colon cancer get before surgery? Some give oral antibiotics alone, tablet antibiotics. Some give a mechanical bowel prep. Some give both, and some give nothing at all. And our question was, are oral antibiotics better than no preparation at all? So comparing those two. There's been a lot of studies in this space. The size of the black line tells you how many studies there have been comparing these. And there's only been one study comparing this in the past. It was published in Spain, a smaller study published in Spain, comparing antibiotics to nothing. But otherwise, it's a question that really has not been asked. Next slide. Sorry, next slide. So we wanted to try first, I think you have to keep pressing through the animation there. Feasibility studies to see if we could use this REACT platform in this way. So we took patients who were having elective surgery at one of our 2 sites and randomized them on the phone to whether they got an oral antibiotic prescription or no preparation at all. Next slide. And we used the same process as the REACT platform. So integrated oral consent, randomized the patient right away on the phone, and then used the NSQIP database to collect all the data for the participating patients. So it was an automated data collection. Next slide. Our primary outcome, I just have to keep pushing through the animation there. Was whether or not we could enroll 80% of patients that were eligible for the trial. So that means that 8 out of 10 patients who were eligible agreed to participate, and then 8 out of 10 of the patients that participated adhered to the arm that they were in. Next slide. So our secondary endpoints were, how many patients were lost to follow-up. We wanted to prove we would lose less than 90% and we could get more than 90% of the automated data collection. And of course, in the larger trial, we're looking at reduction in surgical site infection, length of stay, antibiotic-related complications, and cost-effectiveness and quality of life. So I'll just show you next slide the data from our feasibility study, which was done over the course of just under a year, and we were able to enroll. We had 74 eligible patients present to the hospital, and we enrolled 67 of them. So we had 90% enrollment and 94% adherence. That means that 9 out of 10 patients who were eligible participated, and the majority of those patients, 94%, did keep adhering to the arm that they were in. Next slide. And we had 99% automatic data capture, so we didn't have to look through charts, and we lost no one to follow up. So next slide, we were very excited about this. Our enrollment was sort of tenfold the national average. We had no loss to follow up, and this was the first clinical trial that we were aware of that leveraged this NSQIP data in a randomized clinical trial fashion. And so we thought this was very feasible. Next slide. So the plan was to roll this out across multiple sites, and then of course COVID happened, and we had to rethink how to run this trial, because even though it was very practical, we still had patients coming in for visits, we still had a coordinator involved, and patients still had to go and get antibiotics, etc. And what I think COVID-19 made us realize is that there are definitely some barriers to clinical trial particularly during the pandemic. Coordinators weren't allowed on sites. Virtual care wasn't consistent with some of the regulations, but it also offered an opportunity to rethink how we are doing trials, and that's how we came up with the remote platform. Next slide. So in the remote platform, essentially, all of the REBs for the participating sites, and this is in Ontario, but it's true of other provinces, have centralized REBs, so we can get approvals across many hospitals at one time. The clinical trial coordinator is not on site, so they're a central clinical trial coordinator at an academic site who then interfaces with the patient on Zoom. We actually have a rectal pharmacy, which is one of the larger pharmacies in Canada, delivers the study drug to the patient at their home, and then we have central contracts through NSQIP to collect the data. Next slide. So as discussed, there's a lot of cumbersome process for classic randomized controlled trials, but here with the React platform, we have a significant improvement. Next slide. So here we have integrated oral consent, connecting with a centralized virtual trial coordinator, home delivery of medications, and data collection through NSQIP, as I mentioned. So really a very streamlined approach. Next slide. So essentially, we think that this could actually provide an opportunity for patients at all sites, whether they're small academic sites, whether the surgeons have any experience with clinical trials, to participate in important questions about what is better standard of care and improve post-operative, reduce post-operative complications, improve morbidity. And again, as I said, this is important for cancer patients because having a complication after surgery can significantly impact your cancer outcomes. So that is a trial we are now having up and running at various different sites. And last slide. This is just some of the team members who've been instrumental in getting this trial up and running, and hope to have some results for you in the near future.
Thank you, Rebecca. That was exactly the question I was going to ask, is how quickly can we get results to, which seemingly is a fairly straightforward but highly relevant question.
Mm-hmm. Well, so what we're hoping is this is the first time this platform's ever been done across Ontario. And now we have some collaborators also in other provinces in Canada. But so some of the issues around getting REB, getting the contracts in place, that's what's taking us so much time. So we did receive a grant in the late spring to roll this out to a number of different sites. And since that time, it's really been about contracts and REB. What we're hopeful is that once that contracts are in place, once that REB is in place, we could ask iterative questions. That would be the power of it. So, we can, once we know the answer to antibiotics, we can ask the next question and the next question to improve care.
Yeah. Both of you are clearly incredibly dedicated to doing what matters for patients. From your individual perspectives, I'd be quite curious to understand how some of these beyond the drug innovations, or what we might call very novel new approaches that, for all the right reasons, save overcome many of the barriers in classical clinical trials. What's been your process for engaging patients and or patient groups in terms of defining your work? There's lots written around how to engage patients in a formal clinical trial process. You're kind of on the edges of some of these things. And I think our audience would be very curious to understand how patients have been engaged in your thinking. Were there formal processes? How did you actually know? Did they help to define some of the research questions? Andrea, maybe I could go to you first.
Yes, I mean, I think obviously, patients are critical, right? Without patients, we can't do the research, we can't move forward. So, and I think for us, some of the challenges are just in terms of getting the word out that this trial exists, accruing patients. That's always, incredibly difficult. And I think Rebecca's work really is beautiful in that it will, it's sort of for patients, but also for the researchers in order to be able to have this uniform platform. Because I think a lot of our patients and we struggle with putting patients on the right trial, knowing what trials are available, knowing the results of the trial. As Rebecca pointed out, the patient said, oh, they come to us, oh, I was just on a trial. We can't figure out what trial they were on. I mean, there's just no sort of uniform process that we can utilize, right? Which would save so much time. And then I think even more importantly, help us to identify which patients should go on which trial in a much more streamlined fashion that just saves time. And that's what matters for patients. Like if they're eligible for this trial, if they're the right fit, that's what they should go on. So, something like that is amazing. And we have some of that in the US, but not really. It ends up being a little bit of this no man's land almost at some point. If, okay, we don't have a trial for you at our institution, but they might somewhere else, but we don't have this mechanism built in to be able to best advise patients where to go, even my own patients. So that's a huge challenge for us, but also improving that I think would critically change everything.
Rebecca, do you want to comment on that? Because clearly the application of technology becomes ubiquitous in many ways, doesn't it?
I just wanted to touch on something that Andrea was talking about with the trial of dostarlimab. I think that trial really, when you think about it, it sort of arose from what cancer patients really want. I sometimes ask my cancer patients, what's your blue sky? Sort of what's your ideal outcome? What matters most to you? And I like it because blue sky thinking also sort of means outside the box thinking. And I think it takes courageous patients and investigators to sort of imagine a time when we could have cancer patients cured without long-term toxicity. And really that's what the trial was about. It was about giving younger patients a great quality of life over a long future. And that's, I think, probably where patients are the most important, is sort of asking, like, what matters the most to you and how can we achieve it? And not thinking about incremental gains. I think it's really interesting that patients are much more likely to take a big gamble, like I'll do something if there's a 10% chance of a long-term survival, or what we might call a cure, over a 80% chance of a 5-month increase in lifespan. And doctors are, by and large, less likely to want to gamble on the long shot. So I think it's really important to listen to the patients. Our REACT platform really was created by Dr. Mark Clemens, who's a breast oncologist, because patients said to him, how is it possible that you don't know which treatment is better? I don't remember exactly the context of what treatment, but it struck him as a really important thing that, we should know. Patients deserve to know which treatment is the best. And that's how the platform really evolved.
It definitely, Andrea, is a trial, but at the same time, it's beyond, it's innovative, and it demands, I think, a different kind of approach and thinking across the organization culturally to be able to ask some of these questions. You talked about your team, and I'd be curious to understand how how your team came to some of the conclusions you came to about trying something new, because sometimes we get very stuck in the way we do things.
Yeah, absolutely. I mean, I think, for us, it was sort of this design of the trial and then getting it through the processes that we need to the institutional review board to say, this is medically sound, scientifically sound, and yes, this is safe and okay for patients to do. And then we go to the FDA with the same proposal. So along the way, there was kind of a lot of back and forth and a lot of discussion, with our team and to ensure that everybody was comfortable with this. But as I mentioned, at Memorial, we were already very focused. Our tumor board, our colorectal team, was very focused on this approach of optimizing neoadjuvant preoperative therapy, trying to select patients, a personalized approach to rectal cancer, which is not true for every institution. So I think that certainly helped us, in getting this trial approved and in getting everyone on board that this is something that we were, comfortable doing.
Thank you. It seems as though we've lost Rebecca. We keep losing you. So I think what we're going to do now is just wrap up the session. We're so pleased that I think that both of you are coming back, or we're going to move into a Q&A at this stage, and we'll leave the questions open to the audience. So thank you so much.
Thank you.
Welcome back, and I'm here with Andrea, who's graciously agreed to come back for a Q&A today, which is fantastic because we have so many amazing questions related to your work, and which of course has received so much publicity recently. So thank you again for being with us. So I'll start with the first question, as we only have a few minutes here. And I'm going to read it verbatim. In the rectal cancer study, why was dostarlimab chosen over other immunotherapies that have shown success in colorectal cancer?
That's a great question. So we simply chose dostarlimab because the company that owned the drug at that time, called Tesaro, which has since been bought by GSK, just agreed to take a chance with this trial. We actually discussed it with several other pharma companies because we didn't believe that really it was, this particular drug. We wanted a single agent, we wanted a PD-1 inhibitor, but we did talk to a few other companies and it just was not kind of at their development priority list. So that's how we came about with dostarlimab.
Yeah, it seems very straightforward. So there wasn't a massive analysis.
It's challenging when you have an investigator-initiated study. It's not necessarily very straightforward and very easy to get a drug. So it was definitely a lesson for me that it takes some effort. Yeah.
Again, continuing in the theme in your study, the question here is, what's your response to some of the critics who have actually said that the 100% remission is there because it was a small sample size?
I understand their point. The one distinction that I will make is that the difference here is that it's a small sample size, but it's every single patient. So we actually asked a statistician, what the odds are of getting 100% response in every single consecutive patient, up to 14 patients initially when we presented the data. And it was something like 1 in a trillion, which I could not compute, but it was based on the historical predicted response and what we typically see even in this population. So I think, yes, it's a small study, but the 100%, in every single patient is real.
And I know that based on their conversation, you're intending to expand your results. Maybe you could just reinforce some of the next steps related to the future of the study.
Yeah, absolutely. So we have already expanded the trial to 100 patients in rectal cancer, and we've actually opened a second cohort to all mismatch repair deficient solid tumors, so including esophagus, gastric, pancreas, any tumor that is locally advanced and mismatch repair deficient. Those patients are eligible for the same protocol, 6 months of anti-PD-1 therapy with dostarlimab. And the goal there is to see if we could, of course, replicate these responses, potentially omit some or all part of standard of care, because for many of these tumor locations, for example, stomach cancer, surgery, although effective, is quite morbid. So the goal would be to see if we can have these similar responses where we might be able to also just observe the patients.
Perfect. We only have a couple of moments left, Andrea, but I know you're incredibly proud of the work that you do with the Young Onset Colorectal Cancer Center, and I'd really know that our audience would love to hear more about what we can expect from the centers, some of the efforts, priority efforts that you're focused on, and some of the things that you hope to change, your hopes and dreams for the center.
Yeah, thank you for that question. I do love to talk about it. I think it's so important. So, the center has two goals. One is a clinical goal to help our patients navigate through treatment into survivorship, all of the ancillary support services that are necessary for many of these very young individuals just starting planning their family or have young family or elderly patients that they're taking care of. So there's so many issues that come into play in addition to them having cancer and having to, manage that and of course fight that. And then the second goal, which is equally as important, is the research question. And of course, why is this happening? And the main goal is to identify the patients that are at risk. And in order to do that, we're looking at the tumor biology, we're looking at the early sort of polyp biology to see if there's anything different. We're looking at potential risk factors through a large questionnaire, collecting tissue, collecting stool for microbiome analyses. And then I think most importantly, we're collaborating with other institutions who have also focus their research efforts on young onset colorectal cancer, many of whom we've actually helped establish these centers in the US, including Dana-Farber, but as well as worldwide. In Mexico City, they're opening up a young onset colorectal cancer center. We're collaborating with a center in Israel. And so I think this is a global phenomenon. It's affecting literally almost every country. And so we really need to put our efforts together to try to find out why this is happening and who's at risk.
Any Canadian collaborations on the horizon?
Not yet, but I would love to.
All right. Well, let us help you with that. We would very much enjoy that. Thank you again, for your excellent work. And I'm sorry that Dr. Auer could not join us today, but we'll look forward to seeing more of your progress in the future. Thank you so much.
Thank you so much for having me. Thank you.
Good. We're going to take a 1 moment break as we wait for a couple of our next speakers coming up in the live session. So if we could just take 2 minutes, this would be perfect.